Despite the variability of life on Earth, there are some properties all life has in common. One such property is reproduction. Survival of each species depends on its ability to reproduce. During its lifetime, a parent can have many children. In this way, the population of a species can increase. Under favorable conditions, the population growth of any life form occurs with a characteristic time behavior. This characteristic time behavior of reproduction provided the basis for one of the Viking life search experiments. The time behavior, or rate behavior, that characterizes reproduction can be seen by watching these human cells divide. Under favorable conditions, one cell becomes 2. 2 become 4. In turn, each new cell may also divide. The important characteristic of this reproduction process can be seen on a graph. 4 cells become 8. 8 becomes 16, and so on. The number of cells, plotted against time, follows a curve that becomes steeper and steeper. This ever-increasing rate of growth is a characteristic of every life form placed in a favorable environment. This time behavior, where the rate of growth of a population increases in proportion to the population, is called exponential growth. Exponential growth is a property common to all life forms. Another property common to life is the consumption of nutrients and the production of waste products. For example, every living cell consumes nutrients and gives off end products such as carbon dioxide. The formation of the end products occurs at the same rate as reproduction. Scientists would say both occurrences exhibit the same time behavior characteristics, or reaction kinetics. If an end product, such as CO2, is detected, the time behavior, or rate of its formation, can reveal whether a reproduction process could be responsible. Here is a simple apparatus with which we can measure the time behavior, the reaction kinetics of chemical reactions. Let's begin with the inorganic reaction of sodium carbonate with hydrochloric acid. This flask contains sodium carbonate. Dilute hydrochloric acid is introduced into the collecting funnel. The reaction generates carbon dioxide, which moves through a glass tube to displace a column of water in a graduated cylinder. In the first 30 seconds, 150 milliliters of CO2 is collected. After another 30 seconds, only 15 milliliters of additional CO2 is produced, giving a total of 165 milliliters. The reaction is slowing down. In the next 30 seconds, only 5 milliliters of CO2 is added. We see that for this reaction, in each succeeding 30-second time interval, less end product is formed than in the preceding time interval. This kind of time behavior, reaction kinetics, occurs when a reactant is being used up. Let's contrast this time behavior with a growth process. Ordinary garden soil can demonstrate the reaction kinetics of living, reproducing organisms. Such soil contains numerous microorganisms. The experiment is started by placing about 20 grams of the collected soil into the same apparatus we used in the experiment with sodium carbonate. For this reaction, we will use a nutrient solution that will encourage growth of the microorganisms. The nutrient solution contains sugars, amino acids, vitamins, and seawater, things microorganisms like to eat. Let's see what happens when the nutrient solution wets the garden soil. If the microorganisms reproduce, they will release carbon dioxide. It's 10 hours after the start of the experiment. Time to take the first reading. Hardly any CO2 has collected. Ten more hours have passed. Now we have some CO2. 5 milliliters have collected. After the third 10-hour time segment, there are 18 milliliters of CO2. The reaction is speeding up. These are the data after several more 10-hour intervals. Each measurement shows a larger growth than in the preceding interval. When the volume of CO2 is plotted against time, the curve shows a steeper and steeper slope. This is the characteristic curve we have called exponential growth. This curve provides evidence of the presence of life. There are microorganisms in the garden soil. With enough nutrient solution, the microorganism colony is getting larger and larger. This is Death Valley, California, an area in the Mojave Desert with an extreme climate that is hostile to most life forms. There are few visible signs of life. A sample of desert soil is collected, and a measured amount is added to the flask, just as in the garden soil experiment. The same nutrient is added to this soil, and the amount of reaction product, CO2, is measured at equal time intervals. The gas measurement results are plotted. The exponential, ever-increasing curve shows that microorganisms are even present in desert soil. Life is again detected. What happens if we heat a sample of the garden soil in an oven to 160 degrees centigrade? At this temperature, most microorganisms are destroyed. To this sample of heated garden soil, nutrient will be added. However, even after many days, no gas is produced. There is no growth. The heat from the oven must have killed all the microorganisms in the garden soil. Comparing the plots in each experiment distinguishes chemistry associated with the presence of living, reproducing organisms from the chemistry associated with other types of reactions. The life search experiment based on this principle of exponential growth is called the gas exchange experiment.